Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjon classes. Dear student, we are in a continuation of the chapter that is the cell, the unit of life. And we know it is a basic functional, structural, and functional unit of life. In previous lectures, already we have discussed about the introduction, overview, and the cell theory, and then the difference of prokaryotic and eukaryotic cell in detail, and we have also studied about the cell membrane. But in today's lecture, we are going to discuss mainly about the cell membrane, cell wall, and endomembrane system. Endomembrane system, which is obviously found only in case of eukaryote, that we are going to discuss. And cell wall also we are going to discuss with respect to the eukaryote, because in prokaryote we have already discussed. So first, if we talk about cell wall, in plant you will see the presence of cell wall, but not in the animal cell. And plant cells can be distinguished from the animal cells by the presence of this cell wall, which is absent in animal cells. And this cell wall was first observed by Robert Hooke in 1665 in cork tissues of the oak tree. And it was a Spanish oak. Now, when we see the cell wall, it is considered as a non-living residue structure. It is a non-living residue structure and forms an outer covering of the plasma membrane of fungi and plants. That is present as the outer covering of the plasma membrane, and means it is present outside the plasma membrane or on the outer side of the plasma membrane in bacteria, fungi, and plants are mainly given here. But apart from this, you will see in some bacteria there is a presence of cell wall. In some protists also you will see the presence of cell wall. And in plants you will see the algae, there also you will see the presence of cell wall. So in which tree Robert Hooke observed a cork tissue? This question can be asked, so you should remember that it is the oak tree. Now cell wall is not only gives a shape to the cell, we know it is a protective nature. So besides giving a shape to the cell, it also protects the cell from the mechanical damage and the infection. So it is a protective covering. And cell wall also help in cell to cell interaction. Means if one cell want to interact with another cell, it can interact to the cell wall. Apart from this, it also act as a barrier to the undesirable molecules. It cannot allow the transport or intake of undesirable molecules which are not beneficial for the plant, or which is not required by the plant. So this is the cell wall. If we talk about composition of cell wall, according to NCERT, you need to remember that in algae, the cell wall is made up of cellulose, galactans, mannans. Galactans is a polymer of galactose, mannans is a polymer of mannose, and some minerals are also present in the cell wall of algae, that is a calcium carbonate. While in other plants, if we take, the cell wall is made up of cellulose, hemicellulose, pectin, and some proteins. These are found in the particularly in some plants. Now, the cell wall of a young plant cell, if we talk, the primary wall is capable of growth. Means cell wall is of different type: primary, secondary, tertiary. This way the cell wall is present. And the outermost cell wall, outermost layers of the cell wall is considered as a young cell wall or a primary cell wall which is capable of growth. And primary wall gradually diminishes. It gradually diminishes or reduces as the cell matures. And secondary wall is formed on the inner side of the cell wall. Inner side of the cell wall. So primary cell wall is on the outer side. Inner to that, you will see the presence of secondary cell wall. So the secondary cell wall is formed by the addition of new cell wall material like lignin, suberin, pectin, cutin into the cellulose interfibrillar spaces. Okay, so in the cellulose interfibrillar spaces are there, that the deposition of this material occur, and the secondary wall is laid inner to the primary cell wall. If we talk about some permanent tissue like xylem in plants, you will see that secondary wall exhibit unthickened areas. At some regions, you will see the unthickened areas which are called as pits to support the intercellular transport. So in xylem, if we talk, then some unthickened areas of secondary cell wall are known as which are known as pits, and pits support intercellular transport. Why? Because pits have plasmodesmata, which is a cytoplasmic connection between the two adjacent cells. So that point you should write that pits are the unthickened areas, and within pits, plasmodesmata are present. Then, what are the unthickened areas in the xylem? In the unthickened areas, they are known as pits. Now, when we see the two adjacent cells, then the primary cell wall of two adjacent cells is lined by a middle lamella in between them, and this middle lamella is made up of calcium and magnesium pectate. So if we see here, suppose this is the two cell and this is the primary cell wall. This is the primary cell wall, and here you will see the primary cell wall of the another adjacent cell, and between this you will see the presence of middle lamella here. And this middle lamella is made up of calcium pectate and magnesium pectate. Okay, but in this the amount of calcium pectate is more. Okay, so this is the present between the primary cell wall of adjacent cell B. Remember, this is the cell and its nucleoid. Okay, so now this middle lamella is the first formed layer of cell wall between the primary cell wall of two cells and developed from the cell plate during the cell division. When cell division is occurring, at that time we will study that in the next chapter, that cell plate formation occur and middle lamella is formed. In mature plant, if we talk, the middle lamella is the outermost layer of the cell. Okay, and this middle lamella is found to be get dissolved in the mature fruits. So if we talk about the loose fruits, the fruits, some fruits are ripened fruits are loose. Why they are loose? Because middle lamella is getting dissolved. Whereas if this is a tender fruit or a young fruit, then it is a tight, okay, in a proper shape. Why it is in proper shape? Because the middle lamella is present. It is not giving a loose appearance to the lips. Now, when we see the cell wall, you will see that this is the outermost middle lamella. Then you will see the primary wall. Inner to that, you will see the secondary wall layers. And inner to that, you will see the lumen, as we see the transverse section of the plant cell. So this is the middle lamella. This is the primary wall. This is the secondary wall. Okay. Inner to the primary cell wall, you will see the secondary wall. And secondary wall layers are S1, S2, S3 like layers are found in the secondary cell wall. And inner to secondary cell wall, tertiary cell wall will found between the two, between the sum of the between the sum of the plants. So the middle lamella is composed of calcium pectin mainly. It is also made up of magnesium pectin, and the cell wall and middle lamella may be traversed or passed by a plasmodesmata which connect the cytoplasm of the neighboring cells. So if we talk about the primary cell wall and secondary cell wall, then dear student, first we are going to talk about primary cell wall. Now primary cell wall is thin, elastic, and it is composed of cellulose, hemicellulose, and pectin. As the cell matures, it gradually diminishes or reduces, and it is capable of growth because it is thin and elastic. Then you will see the presence of secondary cell wall. And when we see the secondary cell wall, it is a rigid and thick. It is rigid and thick. And this secondary cell wall is composed of cellulose, hemicellulose, and pectin. But this secondary cell wall is absent in meristem cell, whereas the secondary cell wall is formed on the inner side of primary cell wall. It is formed on inner side of primary cell wall. That is towards the cell membrane. And then inner to secondary cell wall, you will see the presence of tertiary cell wall. And if we talk about the tertiary cell wall, it is present, it is laid inner to the secondary cell wall. And this tertiary cell wall are composed of hemicellulose and xylem. Xylem is a polymer of xylulose, okay? And this tertiary cell wall is not found commonly. It is present only in tracheids of gymnosperm. Okay, there you will see the presence of tertiary cell wall inner to the secondary cell wall. And this secondary cell wall, which we have studied, that can be in several layers like S1, S2, S3. The many layers are present, and we know that middle lamella is a common layer between the two cells. Now, in all this, you will see that cellulose is the main constituent of cell wall, but additionally, addition to cellulose, you will see the presence of hemicellulose, cutin, lignin, suberin, and all this. Now, when we talk about the structure of cell wall, you will see that there are cellulose microfibrils. And when we talk about this cellulose microfibrils, and macrofibrils, they are arranged in a layer to form skeleton of cell wall. And in between these layers, this cellulose microfibrils and macrofibrils are in a layer. And you will see that in between these layers, other substances like Other substances like pectin, hemicellulose may be present, and this form matrix of cell wall. This form matrix of cell wall. Okay, so this cell wall material, that is hemicellulose, pectin, and lignin, are synthesized by the Golgi bodies, and formation of cellulose occur by the cell wall. Okay, cell membrane. Sorry. So here you should remember that cellulose is formed by cell membrane, and hemicellulose, pectin, lignin, etc. are synthesized in plant Golgi bodies or dictyosomes. And you will see that there is a presence of plasmodesmata. Suppose this is a primary cell wall. Okay. Here you will see the presence of middle lamella. This is primary cell wall. And here you will see inner to that the presence of secondary cell wall. Okay, so this is the primary cell wall. Inner to that you will see the presence of secondary cell wall. Now these are the unthickened areas which are known as pits. And in these pits you will see the presence of this connection that is known as plasmodesmata. So plasmodesmata is the cytoplasmic connection between the two adjacent cells. So if we talk about a plasmodesmata, dear student, then you will come to know that it is a cytoplasmic connections between two adjacent plant cells. And this plasmodesmata is a characteristic of multicellular plants. So it is a cytoplasmic connection between the two adjacent cells, and it is a characteristic of multicellular plant, name given by Strasburger. And you will see that there's some endoplasmic reticulum tubules or ER tubules which are known as desmotubules. They help to maintain continuity of cytoplasm. They help to maintain a continuity of cytoplasm. And this plasmodesmata passes through the cell wall and middle lamella of the adjacent cell and connects the cytoplasm of the neighboring cell. So that is all about the what a cell wall. Now last we are going to talk about the functions of cell wall. So if we talk about the functions of cell wall, then it gives shape to the cell. It gives shape to the cell. Then it protects cell. It protects cell from mechanical damage and infection. And it also help in cell to cell interaction. Apart from this, it provide barrier to undesirable macromolecules. So these all are the functions of the cell wall. Okay, so this is the thing about the what a cell wall, and that is all about the cell membrane. Now we have discussed about the cell membrane in the previous class. Today we have completed now about the cell wall. Now we are going to look over the cytoplasm. And when we talk about the cytoplasm, in cytoplasm we are going to discuss the cell organelle. Okay. So now onwards we are going to discuss cytoplasm. Okay. And when we talk about this cytoplasm, then the term cytoplasm given by Strasburger. We remember this term cytoplasm was given by Strasburger for a part of a cell. So term cytoplasm was given by a Strasburger, and it is for the part of a cell which present between the nucleus and cell membrane. And cytoplasm is a part of cell between between nucleus and cell membrane. In this, the liquid matrix except organelle, it is known as cytosol. So cytosol is the term used for the liquid matrix of cytoplasm except organelles. Okay, and apart from tropoplasm, there are non-living inclusion bodies. For that term, deutoplasm is used. So deutoplasm refer to the non-living inclusion bodies. And now this deutoplasm is a non-living inclusion body. So for the organelle as well as non-inclusion bodies, the term trophoplasm is used. So trophoplasm is equal to cytoplasm part containing organelles plus deutoplasm. Deutoplasm means non-living inclusions. So this is the term used for the tropoplasm. Now, apart from this, the another term which is used, that is known as protoplasm. Now, protoplasm is term for the matrix which is present in the nucleus, that is the nucleoplasm plus cytoplasm. Plus sorry. So this fluid part of the nucleus and cytoplasm is combinedly called as a protoplasm. But when it is covered by the cell membrane, it is known as protoplast. So protoplast consists of nucleoplasm plus cytoplasm plus cell membrane, and this protoplast is a cell without cell wall. It is a cell without cell wall. Okay, so it has a cell membrane, but cell wall is not there. And if you remove this cell membrane, then you will have a nucleoplasm and cytoplasm. This combinedly known as what? A protoplasm. Now here the cell organelle are present, and we have to discuss the cell organelle in detail. And these are the living structure and metabolically active structure of the cytoplasm, which are called as cell organelle. And there are several type of cell organelle, but in this first we are going to discuss the endomembrane system, which consists of organelle which have a coordinated function. So let's talk about the endomembrane system. Now, many of the organelles which are membranous or membrane bound, they are considered together as a one unit, that is endomembrane system. Why? Because the functions of this organelle are coordinated. And if we talk about this organelle which have a coordinated function, then this is an endomembrane system, and it includes Golgi complex, lysosome, vacuole, and endoplasmic reticulum. Actually, first is endoplasmic reticulum, then it is connected with the Golgi complex, then to the lysosome, and then vacuole. So first we will discuss about the endoplasmic reticulum. But dear student, apart from this four organelle, mitochondria, chloroplast, and peroxisome are there, which are not included in this endomembrane system because the functions of this organelle are not coordinated with the endomembrane system. So because of this, only four organelles are involved, that is endoplasmic reticulum, Golgi complex, lysosome, and vacuole. This endomembrane system is a single membrane bound system. So we remember all the four organelles have a single membrane around them. In this, first we are going to discuss endoplasmic reticulum. See, when we see endoplasmic reticulum within the cytoplasm of a cell, you will see the endoplasmic is looking like this. And when we see this electron microscopic diagram, you will see that this is the nuclear envelope and this is the nucleus, and outer membrane of this nucleus is coordinated, is connected with this extend, means outer membrane of nuclear envelope extend and form this network of parallel structures. Okay, and this whole is known as endoplasmic reticulum. But near to the nucleus, you will still see that this endoplasmic reticulum have a ribosome over them. So due to the presence of ribosome, this part has some granular structure, rough structure. Due to this part is known as rough endoplasmic reticulum. But the part which is away from the nuclear envelope and near to the cell membrane, that part do not possess a ribosome, and this is known as smooth endoplasmic reticulum. So dear student, if this is the endoplasmic reticulum, K.R. Porter was the first to coin this term and describe the thread-like structure in a cell. So this is the K.R. Porter. Then the studies of eukaryotic cell reveal the presence of a reticulum of a tiny tubular structure. It is a reticulum, it is a network of a tiny tubular structure which is scattered in the cytoplasm, and that is known as endoplasmic reticulum. And it was observed by the electron microscopic studies. Okay. Now, when we see this ER, which is present within the cell, it divides cytoplasm or intracellular space into the two compartments or two distinct compartments. See, let's see previous diagram. In this diagram, you will see that there is a space within the membrane, within the endoplasmic reticulum, and this is the outer side space. So according to this, the ER divides the intracellular space or the space which is present within the cell membrane, complete space, into the two distinct compartments, that is a luminal compartment which is inside the endoplasmic reticulum and extraluminal compartment which is outside the endoplasmic reticulum, that is a cytoplasm, which consists of other organelles. So luminal compartment and extraluminal compartment, two compartments are formed by the ER. So here, if we see the structure of this, you will see that this is a nucleus, this is a rough endoplasmic reticulum, this is smooth endoplasmic reticulum. These tubular structures are known as cisternae, and this is a lumen which is present within the cisternae, that is a luminal compartment, and outside that you will see this is the extraluminal compartment. Okay, now endoplasmic reticulum, if we talk, it is of two types, that is rough endoplasmic reticulum or granular endoplasmic reticulum because it has a ribosome, and smooth endoplasmic reticulum which do not have a ribosome. So it is known as agranular because granular appearance is not same. So when we see the endoplasmic reticulum, it is going to look like this. Okay, and here you will see the presence of what structure like this. So you can see in this structure that outside the nuclear envelope, in nuclear envelope you will see such nuclear pores, then outer nuclear membrane cover this ultimately unwinds and form this endoplasmic reticulum. This have a cistern. So if we talk about the composition of this endoplasmic reticulum or components of the ER, then components of endoplasmic reticulum, if we talk, these components include number one, that is cisternae. And if we talk about this cisternae, these are long flattened, these are long flattened and unbranched units and unbranched units arranged in a form of stack. So this cisternae are like this. They are unbranched and long flattened structure. Okay. This cisternae bear ribosome. So it is a main compartment of rough endoplasmic reticulum, mainly present in rough endoplasmic reticulum. Okay. Now after this, second ones are the tubules. Second component is tubules. And when we talk about these tubules, these are irregular, often branched tubes, often branched tubes bounded by membrane. Now tubules can be free or associated with the cisternae. So tubules may be free or associated with cisternae. So this is the what a structure, and tubules may bear ribosome or not. They may bear ribosome or may not. It can have a ribosome present over this or not, and it is mainly present in or mainly found as a component in smooth endoplasmic reticulum. And if we talk about these tubules, these are a branched structure, so they are look like somewhat this. Okay, and ribosome may or may not be present over them. And the third component, which you can see, that is vesicles. These vesicles, if we talk, they are oval membrane bound structure. They are oval membrane bound structure, which are a part of this component of ER. And now these vesicles are released from the terminal part of the endoplasmic reticulum, and it contains some proteins. Okay, and that proteins can be what, subsecretory proteins or some immature proteins which are synthesized by cisternate or ribosome, that is taken away by these vesicles. So this is the structural component of ER, and when we see the structure of this ER, the structure of ER is like Golgi body, but in ER, cisternae, vesicles, and tubules are isolated in cytoplasm, isolated in cytoplasm, and these do not form network. Okay, so this is the thing. And this endoplasmic reticulum is widespread in cytoplasm. So because of this, ER is also termed as a system of membranes. Endoplasmic reticulum is widespread in cytoplasm, so often termed as system of membranes. Okay, so that is what about the components of the ER. Now we talk a type of ER, and these two types of ER are there: rough endoplasmic reticulum and smooth endoplasmic reticulum. First, we are going to talk about this rough endoplasmic reticulum, and we discuss this rough endoplasmic reticulum. Then on their surface, you will see the presence of ribosome. And this RER is frequently observed in the cells which are actively involved in photosynthesis, because ribosome has a function of photosynthesis. So whichever the cell which need a more protein or that need to secrete more protein, that have a large part of RER, or RER is frequently observed. Okay, so such a way the arrangement is found. Now RER is extensive and continuous with the outer membrane of the nucleus, means it is a coiled structure but is originated from the outer nuclear membrane. So when we see rough endoplasmic reticulum, it is like this, which has black colored dots. You can see they are the ribosome, and that is the membrane. Okay. So if we talk about rough endoplasmic reticulum, this is also known as RER, or granular endoplasmic reticulum. Why they are known as granular? Because it bear ribosome, and this ribosome are of ATS type. So ATS type of ribosome are present over this. And this, suppose this is the ER cisternae, and over which if ribosome is attached, then ribosome is attached through its large subunit. So if it is ribosome, this is the 40S subunit, and this is a 60S subunit. And 60S subunit, you will see the presence of here these two glycoproteins. So two glycoproteins are there. So here we can write that ATS ribosome binds by to ER membrane through larger subunit with the help of with the help of two glycoproteins on the which are present on the surface of ER. They are known as riboforin 1 and riboforin 2. Okay, so this riboforin 1 and 2 are present on the membrane of surface of rough ER, and through this they are attached to the large subunit. And obviously it is mainly composed of cisternae, and it is abundantly present in the cells which are actively engaged in the protein synthesis and secretion like liver, pancreas and goblet cells. So they abundantly occur in liver, pancreas and goblet cell. Now, let's talk about the functions of RER. Functions of rough endoplasmic reticulum. So if we talk about the functions of rough endoplasmic reticulum, dear students, they have ribosomes, so it is mainly involved in protein synthesis, which is the main function. Okay. Apart from this, it is known that it bear enzymes in the region of pores for modifying polypeptides synthesized by attached ribosome. An example is glycosylation. Okay, so the functions of ER is protein synthesis. Apart from this, it is also carrying out glycosylation, that is a modification of the proteins which are synthesized by the ribosome attached to the RER. It also provide if we talk about other one, it also provide enzyme precursor for the formation of lysosomes by Golgi complex. And it is known that the smooth endoplasmic reticulum, that is SER, developed from RER because the RER continue and form a SER toward the what we can say the plasma membrane. Now, apart from this, next is the SER, that is a smooth endoplasmic reticulum, and it does not bear the ribosome. So, in this, the absence of ribosome gives endoplasmic reticulum a smooth appearance. So, it is known as smooth endoplasmic reticulum, and it is a major site for the synthesis of lipids and steroids. Okay, it is involved in mainly what a sterol synthesis. Okay, so that is what smooth endoplasmic reticulum. In animal cell, lipid-like steroidal hormones are also synthesized by the ACR. So just we have understood that steroidal hormones are synthesized by the ACR in animals. And in animals also they are specifically synthesized in some of the adipose tissue, muscles, glycogen storing liver cells, and some of the adrenal cortex cells. There you will see the particular synthesis of these tubules. When we see the structure of SER, which is present towards the plasma membrane, it looks like some of this. You will not see the presence of ribosome, but you will see the cisternae, you will see the tubules, you will see the vesicles, but some structures. But ribosome are not there. So if we talk about this SER, SER is free from ribosome and the ribophorins. Now, before understanding the functions, okay, this SER or smooth endoplasmic reticulum is also known as agranular endoplasmic reticulum. Okay, it is known as agranular endoplasmic reticulum. And it does not bear ribosome, obviously, is mainly formed of vesicles and tubules. It is known that SER is mainly formed of vesicles and tubules because if cisternae are present, they are also present in very less amount. Okay, and it is involved in synthesis of lipid, steroids, and glycogen. Pores are absent in SER. And due to absence of pores, the material synthesized by SER do not pass into the channel. They are not going to pass into the channel. And this SER is often peripheral. So it may be connected with plasma lemma and it developed from ARIA. Now, one more thing. Okay, so this is the what particular different component with respect to the SER. Now we are going to discuss the functions. And if you talk about the functions of SER, we have studied that it is involved in mainly a lipid and steroid synthesis that we have written already. But we have to note down some specific functions of the SER, and we have to note down some of the specific functions. It also carry out, see, it carry out synthesis of glycogen as well as hydrolysis of glycogen as well as hydrolysis of glycogen in liver cells. Okay. Apart from this, it is known that it stores calcium for release during muscle contraction. Apart from this, it is known that it take part in detoxification of toxic chemicals with the help of cytochrome P450. And it is known that SAR help that the synthetic products of RER pass on to Golgi bodies, pass on to Golgi complex through SER. Okay, so this is all about what? Particularly we can say the functions of SER. And whenever the any protein which is synthesized by the RER, it is passed on to SER and it is packaged in the vesicle and exported through the vesicles. So the vesicles which take away this immature proteins, or we can say the proteins which are synthesized by endoplasmic reticulum, these vesicles are known as secretory vesicles. Okay, secretory vesicles. So this is all about what? SER and RER. Now after this, we are going to discuss a modification of some ER. Modifications of ER or endoplasmic reticulum. When we talk about modification of ER, first we talk about the sarcoplasmic reticulum. And when we talk about the sarcoplasmic reticulum, it is a smooth ER. It is a smooth ER of skeletal and cardiac muscle. And this SER, this sarcoplasmic reticulum, store calcium and energy compounds required for muscle contraction. So that is a sarcoplasmic reticulum. Another modification is microsomes. If we talk about this microsome, These are pieces of ER, and this pieces of ER with with associated ribosomal particles. And it is known that these microsomes, if we talk, they are obtained by obtained by fragmentation and high speed centrifugation. High speed centrifugation of cell. Actually, they do not exist as such in living cells. They are not found as such in the living cell. They are produced by the fragmentation or high speed centrifugation. But scientists used this microsome for the in vitro protein synthesis or artificial protein synthesis. For this, they can use this particular. Okay, so this is the what some of the particularly we can say functions and modifications of the ER. So who first observed the cell wall? We know that it is by the Robert Hooke. Outermost layer of cell wall, it is obviously middle lamella. Plant cells are connected by channels through their walls called plasmodesmata. An extensive network of tiny tubular structures scattered in cytoplasm continuum with the outer membrane of nucleus is endoplasmic reticulum. And the following cell organ not included in the endomembrane system, obviously, lysosome, Golgi complex, and ER are included, but mitochondria is not included. Okay, so this is all about endoplasmic reticulum or in detail. So dear students, let's talk about the Golgi apparatus. When we talk about this Golgi apparatus, it is also known as Golgi body. Okay, or it is also have some other names. So this Golgi apparatus, if we talk, it is first observed as a densely reticular structure near the nucleus by the Camillo Golgi in 1898. So he observed first time this near the nucleus. And if we talk about this Golgi apparatus, it consists of many flat disc-shaped structures called cisternae. And this cisternae has 0.1 micrometer to 1 micrometer diameter. And this cisternae, which are a tubular structure, which are stacked parallel to each other. When we see about this cisternae, which is one of the component, actually it has a three parts: cisternae, tubules, and vesicles. This cisternae are an important one, and they are present in a Golgi complex. And the structure look like this. See, this is the cisternae, and this cisternae look like this. Okay, and here you will see that the cisternae are there, these vesicles are there, these vacuoles are there. The Golgi cisternae are concentrically arranged near the nucleus with a distinct convex cis or the forming face and the trans or maturing phase C. This is the structure of Golgi body, which has this tubular structure which are known as cisternae. Okay, cisternae. Now see, this is the cis phase or receiving side of the Golgi apparatus, which is towards the nucleus. It is towards the nucleus. Okay, whereas this is the convex phase, whereas this is a trans phase or concave phase, and this concave phase is the shipping side of the Golgi apparatus, or it is a trans phase, or it is a maturing phase. So if any protein which is coming from the smooth endoplasmic reticulum with the protein, that will come and bind here to this cis phase, and this load this protein to this cisternae, then the cisternae will pass on this to the trans side, and up to moving to the trans side, the modification of this protein will take place, and this protein is going to be mature. Okay, so this is the thing which occur in case of the Golgi apparatus. So Golgi complex carry out a functioning of a packaging of material to be delivered to the intracellular target, means that are required within the cells, and or to the secretory outside the cell, means whatever the protein which is received from the endoplasmic reticulum is getting matured or packaged in the Golgi complex, and it is given to the intracellular target or within the cell or to the outside, means it is secreted outside. So this is the thing. So materials to be packaged in the form of vesicle from the ER fused with the cis phase of the Golgi apparatus. Dear students, cis phase is nothing but which one? We can say the convex surface of cistern, okay, and which is towards the nucleus where it receives the vesicle from the ER and then move towards the maturing phase, and from this it is released or released with the packaged proteins which is matured. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. Why? Because whatever the protein which is made by the endoplasmic reticulum, it is processed, packaged, and modified in the Golgi complex. So proteins are synthesized by the ribosome in the endoplasmic reticulum and further modified in the cistern of Golgi apparatus before they are secreted from the transphase. In plants, if we see, Golgi apparatus involves the synthesis of cell wall material and also plays a role in formation of cell plate during the cell division. Okay, so in plant, if we see, this Golgi apparatus involves the synthesis of cell wall. It provides material for the cell wall and also play a role in formation of cell plate during the cell division. So if we talk about the Golgi apparatus, it is the important site for the formation of glycoproteins, it is an important site for the formation of glycoproteins and glycolipids. So these glycoproteins and glycolipids are synthesized or formed by the Golgi apparatus. And this is the Golgi apparatus structure. Okay. So now if we talk about Golgi apparatus in more detail, we come to know that this Golgi body is also named as number 1 lipochondria. Why it is known as lipochondria? Because it is rich in lipids. And second name is dictyosome, which is a term used for the plant Golgi body. Okay, so this is the name. Apart from this, it is also called as Dalton complex and fourth apparato reticulare. So this is the different names for the what a Golgi complex. Now it is known that the cytoplasm which is surround this Golgi body, it is known as zone of exclusion. So what is zone of exclusion? It is a cytoplasm surrounding Golgi body which have fewer or no other organelle. And this zone of exclusion, which is surrounding cytoplasm to the Golgi body, which has fewer or no other organelle, it is also called Golgi ground substance. Okay, so this is the what particularly we have seen. Now, if we talk about a structure, Golgi complex is made up of three parts. Number one is cisternae, and these are the disc-shaped structure. We have already understood the points about this. They are in a varied number. The convex phase is known as cis phase or forming phase, where a vesicle from the ER get fused, and the concave phase is the trans phase or maturing phase from which the particularly modified protein get released from the vesicles. And the cis and trans phases of organelles are entirely different but interconnected. We remember in system A, one important point is that the cis and trans phases of organelle are entirely different. Are entirely different, but interconnected. The another component or part is tubules. We have studied in the ER that cistern are unbranched, but here also in Golgi also cisterns are unbranched, and these tubules are obviously they are a branched structure, branched and tube-like structure associated with cistern. And third is the vesicles. So these are the oval structure which contain modified protein. So these vesicles will be considered as a mature vesicle because this has a mature protein. So this is what about the structure part and the how this is made. Now after the structural component, we have to talk about some more points, and it is that Golgi occurrence we have to note down. If we talk about the occurrence, Golgi apparatus or complex is absent in Prokaryotic cell, obvious, that we know. And apart from this, it is known that it is present in all eukaryotic cell except sieve tube of plants, sperms, or antherozoids of bryophytes and pteridophytes. And apart from this, RBC of mammals. Okay, so these are the regions or the structures of the cells in which you will see the presence of this Golgi complex. Now this is what about the particularly the structure and occurrence. Now we are going to talk about the functions. So when we talk about the functions of Golgi complex, the functions are: number one, cell secretion. Cell secretion is the chief function of Golgi body. So cell secretion means export of macromolecules, and this export of macromolecules is carried out by Golgi body, and it is the chief or main function of Golgi body. And when we talk about this secretion involves three steps. So this secretion, it is known that it involves three steps. Number one, we can say A, that is to receive the material from ER through cis phase. Okay. Then second step is modification of this material by glycosylation or glycosidation of proteins and lipids to form glycoproteins and glycolipids. So proteins are undergo glycosylation. These proteins undergo glycosylation and form a glycoprotein. Same way, the lipids undergo glycosidation and form a glycolipids. These structures are formed. And then third, they are packaged into vesicles. And these vesicles, which have a packaged or modified protein that are pinched off from the trans phase of Golgi body and discharged outside the cells. So packaged into vesicle and then secreted outside. Okay, so this is the cell secretion. So here we can understand that all macromolecule are to be sent outside, they move through Golgi body. So we can say that all macromolecules which are sent outside the cell move through Golgi body, so termed as middlemen of cell, or we can say director of macromolecular traffic in cell. Okay, so these things are done by this Golgi apparatus. Okay, after this, second function is formation of lysosome. So lysosomes are formed by the Golgi body. It is not a only function of the Golgi, but Golgi carry out this function in association with the ER. So it is known as collective function of Golgi body and ER. Third function is synthesis of cell wall material. That is a polysaccharide synthesis. And fourth is cell plate formation. Okay, so this cell plate formation is also carried out by this. And fifth is we remember that this cell plate is formed during the cell division in plant cell. And when we talk about the sperm, that is male gamete, in male gamete acrosome is there. So formation of acrosome during spermatogenesis. That is also carried out by this particular, we can say, the Golgi complex. So this is all about the Golgi complex. Now we are going to talk about the third organelle, which is a part of endomembrane system and which is in a coordination with the endoplasmic reticulum and a Golgi body. So if we talk about this lysosome are rich in, they are actually rich in some of the hydrolytic enzyme, which we call a hydrolysis. Okay. Now, the lysosome is obviously a membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. So, if the vesicle is coming from the trans phase of the lysosome, trans phase of the Golgi complex, and if this vesicle has some hydrolytic enzyme, then we can call this vesicle as a lysosome. Okay, so that is the thing. So the isolated lysosomal vesicle are found to be very rich in hydrolytic enzyme or hydrolases, and hydrolases are capable of digesting carbohydrate, protein, lipids, nucleic acid, all this. Because this hydrolases, if we talk, they are also known as acid hydrolases, and this acid hydrolases which are found, they are lipases, so they break the lipid. Okay, this acid hydrolases which are present in the lysosome, they can be also proteases. So if it is proteases, it is going to cleave the proteins. Then carbohydrates, then they are going to cleave the carbohydrate. And nucleases, so they can cleave the nucleic acid, that is DNA and RNA. Okay, so this hydrolases are found in this lysosome. So just we have discussed the example of hydrolytic enzymes. If somebody asks, then they can be lipases, proteases, nucleases. So this is the, you can say, the structure where you can say this is the plasma membrane, okay, bilayer, and hydrolytic enzyme mixture is present within this. Glycosylated membrane transport proteins can be there. So such anatomy of lysosome is seen. So this lysosome, if you see its structure, it is a single wall membrane, means single membrane bound structure. So the hydrolytic enzyme are optimally active at the acidic phase. So all this hydrolytic enzyme, either it is a lipases, proteases, nucleases, carbohydrates, whatever it may be, they are active at the acidic pH. And if we talk the acidic pH is somewhat around pH 5 is found. Actually, it is known as 5.65 is found in the this lysosome. Now, this lysosomes sometimes help in autolysis. So, under starvation condition, lysosome digests cellular content by releasing hydrolyzing enzymes. Means if a nutrition is not getting, cell is not getting nutrition. So, what it will use? It will use its all content of the cell, and that content of cell is used by this lysosome. Then it is known as autolysis. So, under starvation condition, you will see that lysosome digests the cellular content by releasing hydrolytic enzymes and cause the death of cell. That is known as autolysis. Okay, so now we have to note some more things which are peculiar with respect to the lysosome. So it is known that this lysosome are reported all animals. This lysosome are reported in all animals, all animals cell except mammalian RBC. Now this lysosome are less frequent in higher plants, but they are more frequent or large number in fungi. Now we know that this lysosome are covered a bag-like structure which is covered by a single unity membrane, and this lysosome, it is known that They are filled with 50 different type of 50 different type of digestive enzymes, and they are termed as acid hydrolases for the digestion of all type of macromolecule. In the lysosome, you will see that acidic pH is there because active H plus pumping is take place within the lumen of lysosome. So due to this, the pH is acidic. pH is acidic due to active pumping of H plus in lumen of lysosome. Then it is known that lysosome are highly polymorphic cell organelle, are highly polymorphic cell organelle. So we can say they show polymorphism, and polymorphic means it have different physiological state, or we can say they are of different type. Okay, so this is what about the lysosome. Now we are going to discuss the type of lysosome. So when we discuss the type of lysosome, dear student, first type is primary lysosome, or we can say storage granule. So it is a primary lysosome or storage granule. So this lysosome store inactive enzymes. So it has inactive enzymes. So lysosomes with the inactive enzymes are considered as a primary lysosome or storage granules. Now after this, there is another type that is known as secondary lysosome or digestive vacuole or heterophagosome. So if we talk about this heterophagosome or secondary lysosome or digestive vacuole, these are formed by fusion of primary lysosome and phagosome. So we know that phagosome is formed by the endocytosis, and phagosome have a food material. Okay. Now when this lysosome is going to bind with this, that is primary lysosome. and this is a phagosome. So when phagosome bind with this primary lysosome, this primary lysosome has enzymes which can digest with this material of the phagosome. So now these enzymes are going to be active. So combinedly they will form having a food material which is getting digested and the enzymes, then it is a secondary lysosome. After this, in secondary lysosome, the digestion keep on going, but at last some undigested matter remains in this secondary lysosome. Then it is known as third type of lysosome, that is residual bodies. So residual bodies are the lysosome containing undigested material. Containing undigested material. And this residual bodies, if they have undigested matter or waste matter, then it should be released outside. So they are eliminated by exocytosis. Then fourth is autophagic lysosome or autophagosome. And this autophagic lysosome or autophagosome, if we talk, then this is the lysosome containing cell organelle to be digested. They are also known as autophagic vacuole. Now what they do, suppose this is the mitochondria which is dead, okay, and we need to digest it properly. Okay, suppose this is the mitochondria. Let's draw it properly first. Suppose this is the mitochondria. And if we want to digest it, then the primary lysosome will come and they will come around this cell organelle, and this primary lysosome which surround this organelle, then they fuse and then release enzyme over organelle and digest the organelle. So because of this, they are known as autophagic vacuole. Okay, so this is the what different forms of the lysosome. So if lysosome are present in different forms, so we call it as a polymorphic organelle. Now after this, we are going to talk about the functions of lysosome. And when we talk about functions of lysosome, dear students, the first function is intracellular digestion. That is the digestion within the cell. And it is of two types, heterophagy. Heterophagy is about digestion of foreign material. And obviously the foreign material that come within the cell, which is going to be digested by the lysosome, that is obviously taken within the cell by the phagocytosis or pinocytosis. and the B is autophagy. Now this autophagy takes place during the starvation of cell. During starvation of a cell or a deficient food supply. So when there is a starvation of food, then the digestion of old or dead cell organelle take place. And when a old or dead cell organelle get digested, then it is known as autophagy. Apart from this, they also carry out extracellular digestion. And in extracellular digestion, it is known that if we talk here, if we take an example of lysosome of osteoclast, that is a bone eating cells dissolved unwanted part of lysosome by this osteoclast activity. Okay, so this is the extracellular digestion. And after this, the third we are going to discuss, that is cellular digestion. It is a third function, and this cellular digestion is known as autolysis. So sometimes it is known that lysosome burst to dissolve cell completely of a cell, burst to dissolve cell completely. And when they dissolve the cell completely, or they have a capacity to dissolve the cell completely, so lysosome called as suicidal bag of cell. They are known as suicidal bags of the cell. And obviously, by this autolysis, the old cells are also getting removed. If we take one more example here, then cathepsin protein of lysosome. It is known that it digests tail of tadpole of frog during its metamorphosis. So metamorphosis is a stage during which the larval stages are converted into the adult one. So during this, they digest the particular tail. So these all are the functions of the lysosome, and that is all about the what? Lysosome. Now we are going to last organelle of this endomembrane system, which is in coordinated function this with this Golgi complex and the lysosome. So if we talk about vacuole, the vacuole is the membrane bound space which is found in the cytoplasm of most common to plant cell. So it is found in plant cell. It is obviously membrane bound, single membrane bound, and it is a space. So when we talk about the plant cell have a central large vacuole, and this central large vacuole, if we talk, is occupy a 90% volume of the cell. And the membrane of this vacuole is known as tonoplast. It is known as tonoplast, which is a single membrane around the vacuole. Now, what is this tonoplast? So we have just studied the vacuole is bounded by a single membrane that is called a tonoplast. When we talk about this vacuole, the vacuole has a vacuole sap, and this vacuole sap is composed of water, some of the excretory materials, some of the metabolic byproducts, and some waste material. Okay, obviously it is E is not there, it is a byproduct, and some of the waste material which are not useful for the cell. All these are packaged within this tonoplast and covered by a single membrane, that is a tonoplast. If we talk about the some plant, their vacuolar sap also contains a pigment, and the pigment which can be present, that is known as anthocyanin. It is a water soluble pigment, so it is present within the, it can be present within the vacuole, and it impart color to the plant part. And this is the structure of anthocyanin. No need to remember the structure. Now, when we see this vacuole of the plant, it occupies the 90% of the volume of the cell, and it play important role in osmoregulation. Okay, osmo means water balance. If we talk about the tonoplast, it is a selectively permeable membrane, so it facilitates the transport of number of ions and other materials against a concentration gradient into the vacuole. Means it is pushing within the vacuole against concentration gradient by the use of energy by the activity of transport. Like this, it work against concentration gradient. Hence, their concentration significantly higher in the vacuole than in the cytoplasm. So these all waste material, excretory products, all these are more in vacuole than the cytoplasm. So what is the important role of vacuole? Just we have discussed osmoregulation, or osmoregulation is the regulation of water. Now, when we talk about vacuoles, they are of three types. Vacuole are of three types. One is gas vacuole, another one is the contractile vacuole, and another is the food vacuole. If we talk about the amoeba, in amoeba you will see the contractile vacuole. And this contractile vacuole play an important role in excretion of unwanted material, or the, we can say, the osmoregulation. So this is the picture of amoeba, and it has a pseudopodia which engulfs some material, and along with this it has the ability to remove the excretory materials. Then in many cells, as in protist, you will see the presence of food vacuole, and which is formed by the engulfing of the food particles. So here the oral groove is present in this paramecium, which particularly engulf the food and form a food vacuole. You can see this is the paramecium, and in this paramecium you will see the presence of food vacuoles. Okay, so this is all what about the, we can say, the vacuole. So engulfing of food material is also one of the important mechanism. Suppose this is a pseudopodia, this is a we can say amoeba, then amoeba, if you talk, it is a small organism, and this has a pseudopodia. So amoeba approaches a food and it forms a pseudopodia around the food particle, and eventually surround the food, and the food becomes enclosed in the food vacuole, which comes within the, what, this is the, see, this is the food particle, and this is the pseudopodia. Now this engulf a food material, and then it form a food vacuole. So digestive enzyme break down the food, and the nutrients diffuse into the cytoplasm and used wherever they are required within the, particularly we can say, the part. So this is what about the vacuole which we have understood. Now, dear student, this is all about the four different, we can say, organelles which are in a coordinated function. Okay, so that is all about the endomembrane system. Now let's talk about some of the last MCQs. The cell organelle help in food digestion process in plants. So obviously digestion is carried out by the lysosome, that is intracellular digestion. Now hydrolytic enzymes are abundant, and obviously for intracellular digestion hydrolytic enzymes are required which are present in the lysosome. An old plant cell is characterized by, if we talk about old plant cell, It is obviously characterized by a presence of large vacuole which occupy 90% volume of the cell. The vacuole contains, if we talk about the vacuole, and if we talk about composition, vacuole contain one pigment, water soluble pigment, that is known as anthocyanin. Densely stained reticular structure near the nucleus. Obviously, densely stained near reticular structure near the nucleus is nothing but a Golgi complex. Okay, so this is all about the endomembrane system and the cell wall that we have covered in this lecture. Okay, I hope you understood very well. Thank you.